a whole bunch of stuff like I had planned this weekend because I just really didn't sleep well, to say the least. But I did this morning. I started working on it, but I finished it, and it should be visible. So I extended the deadline for the agreement. Remember, this is for face-to-face students, not online students. But you do need the lockdown browser. There was like 5 or 6 of you guys that completed it before I made the last changes this morning. So if you have a comment next to your grade, it says, I gave you another temp, please redo it. And I did do as promised, and this is for both my online and face-to-face classes. If you look in the modules, I turned on the practice version of test 1. So you could practice taking it if you wanted. I don't know why when I click it's not going where I want it to go. Oh. It says I'm connected to the internet, so it's just slow. Okay, but under the module it says tests. You'll see practice exam 1 listed. It doesn't seem to want to go there right now, so we'll just move on.
Did it merge our grades?
I had not checked that. He said for me to check today, and if it wasn't fixed, to contact the Wiley Plus guy. So they were asking about the grades merging from Wiley Plus to Canvas. And remember, it's because I accidentally turned on, or you guys convinced me to turn on that link I wasn't supposed to turn on because of how I set it up in my class. It's all good. So they were supposed to sync my grades. So if that isn't done by the end of the day today, I will contact them and make sure they do that. The good news is if you can see whatever grade you have in Wiley, I have access to that too. And so I will make it match at some point if I have to. That's the agreement I just talked about. No, that's in Canvas. You have to download Lockdown Browser. You're meeting with me after class, right? We'll take care of it then. Okay, because we need to move on. Okay, so I even marked where we left off. Of course, now I'm trying to find it. Oh God, where did it go? Huh. There it is. See? Look, see, I marked it. Okay, so we were talking about replication, right, and how important the structure of DNA is. So now, just a little quick, very quick review of transcription. And those of you guys that printed out slides, you'll notice I've even deleted some more slides that are just stuff we don't need to go over. Or I'm not going to spend the time to go over because they won't link up. Okay, because remember all this stuff at this point, we're still on what you should already know before you walked into this class. A segment of DNA, right, can be transcribed into a product, right? The most common one is messenger RNA, right? And there's a few differences between RNA and DNA, right? But they're both nucleic acids, right? One of the major differences is the sugar component. So deoxy stands for the fact that this ribose sugar is missing an oxygen that the ribose sugar has that's found in RNA, right? So that's the difference, DNA versus RNA, is in the difference of the sugars. Then there's one other difference. Instead of having thymine as a nitrogenous base that we abbreviate T, that's substituted for uracil that we abbreviate U. So if you were looking at a code, right, and you saw A, T, C, G, C, C, T, T, A, you know that's DNA or RNA? DNA, why? Because of the T's, right? So if I say AUG, AU, GGCC, G, what am I speaking in? RNA, because I said what? Use. Okay. Another major difference is DNA is double-stranded, where RNA is single-stranded. Same thing, there's an initiation of the process, there's a process of making the RNA, and then of course the process is terminated, is ended. We're not going to go into all the little particulars, right? The most common form of RNA we think of as messenger RNA that carries that message from DNA, right? That message is carried to the ribosome, which is itself also made out of a special type of RNA, known as ribosomal RNA, abbreviated little r RNA. Then you also have transfer RNAs that help transfer amino acids. We talked about this last time, right? To that complex of the messenger RNA, the ribosome, then the transfer RNAs come in. And that brings us to our next process, which is translation. So here, we're actually going to a different type of molecule, right? We're going from RNA to, what do we make in translation? What do ribosomes make? Proteins. What are proteins made out of? What are their repeating subunits? Amino acids, right? So that process is how we make proteins. We've got to first make the transcript of RNA, messenger RNA. From the DNA, we've got to make the transfer RNAs too. We've got to make the ribosomal RNA and the protein to make up that ribosome. There's, of course, a start to the process, an initiation. There's an elongation as we form that protein, and then a termination to the process. This process costs a lot of energy, so it is very highly regulated, right? We're not going to make proteins we don't need for sure, right? Because that'd be a waste of energy. And energy is precious, right? We don't want to waste it. So this was the one process that I definitely wanted you guys to review, right? So the animation does a good job at that. So I had even posted the link to that for you guys. So this starts getting into the stuff you may have not covered in your other class, and I wouldn't probably expect that you would have. But maybe you did in your biology classes. But this directly links to what we're going to study in this class, which is genetic change mutation. So within the messenger RNA, there is a grouping of 3 nucleotides. This grouping is referred to as a codon. This matches and it's complementary to another 3 nucleotide sequence within the transfer RNAs. That's called the anticodon. It's opposite, of course. It's complementary to the codon. Each transfer RNA, based on its anticodon, has an amino acid attached to it. As it comes in and binds, it transfers that amino acid onto the growing chain to form that protein. What they're not showing in this picture is the ribosome that's facilitating this, right? The cool thing about this is we know the code. This is like the secret decoder ring, right? for RNA to protein. So if you take this into consideration that we're only looking at 3 at a time, right? That's a codon. That codes for a single amino acid. And there are 4 different possibilities of nitrogenous bases. You end up with 64 different combinations. How many different amino acids do we have naturally occurring? 20. I just said we had 64 codes. We've got more code than we do amino acids, right? So do we have some repeats? Yeah, when you look at it a little bit closer, right, notice that just as long as you have a C and a U, it doesn't matter what's in that 3rd position. It's going to be leucine. Where for some of them, like look at this first one, if it's UUU, it's going to be phenylalanine, but if you change it to UUA, that's going to be leucine. So probably the most important part of the codon is the first two letters, right? And sometimes that third. That third is sometimes referred to as the wobble. You can have some changes there, and we're not really going to change the amino acid depending, right? Do I expect you guys to memorize this code? Oh God, no, I don't even know it, right? You can look it up. But understand how it works is what we want to try and get at. So you'll notice that 3 of them don't code for amino acids. It says stop. Because guess what the ribosome is going to do when it gets to that code? It's going to stop, right? Apparently he needs a stop sign, right? Otherwise it just will keep going. There's one special codon. In most texts it's highlighted in green. It codes for a specific amino acid, methionine. Notice no other ones code for that amino acid. That codon has a special due purpose other than coding for methionine. Anyone know what it is? It's the start codon. Hence why a lot of times it's coded in green, right? Green for go. That's when you'll see a lot and you'll memorize if you get to my level because you see it so much. But again, I'm not going to make you memorize that. But because of that, most bacteria proteins start with methionine because they don't really modify their proteins and stuff as much as eukaryotes do after translation. So we know the code. It's been worked out. Interestingly, the messenger RNA doesn't need to be an exact match to the anticodon. So you would think, right, that we would have like 64 different transfer RNAs. But sometimes that transfer RNA, again, as long as the first 2 match, it's going to put leucine there. So they don't have to wait for the exact match transfer RNA to come along. So there's that start codon. Before that, there's actually a segment within the messenger RNA where one portion of the ribosome actually attaches, and you'll notice that it's actually the RNA component, the ribosomal RNA, which of course has RNA, matches up with the complementary sequence. This is found in all the messenger RNA. It has this sequence so that the ribosome can attach. And then the other portion of the ribosome is going to attach, right? But it's not going to start until right here at the start codon, actually translating those codons into amino acids to build the protein. So this name, of course, this is the name of 2 scientists that discovered this phenomenon. But what's interesting in bacteria is that because they don't have a nucleus, the messenger RNA comes off the comes off the DNA right there, right? So the RNA polymerase is making RNA right off the DNA, right? And it's just going and going and going. And it'll just keep going. It'll make this really long transcript that has multiple genes on it. So notice it has a starting point and a point for the ribosome to attach, and then it reads, and then there's the stop. And then here's another point where another ribosome can attach. And notice multiple ribosomes will attach at the same time, right, and make a whole bunch of these proteins. So they made 3 separate proteins from this just one long transcript. And so they refer to this as coupling of transcription and translation. This doesn't happen in eukaryotes. Eukaryotes make their RNA in the nucleus and it travels to the cytoplasm or the endoplasmic reticulum where ribosomes attach to it there. And they just do a single gene. In addition, inside the nucleus, they will chop up that messenger RNA and modify it. Why would you want to make protein number 1, 2, and 3 all at the same time? Why would you maybe link these 3 together? What do proteins do? What do proteins do in our cells? What are they? In our cells even. Bacterial cells, our cells, doesn't matter, cell. Why do we make proteins? Why do we make so much protein?
For energy.
Energy. How do we get energy? Right, how do we form ATP? Huh? No. How do we get there? How do we even make ATP? Citric acid cycle. So in that cycle you make citric acid. How is it that you make citric acid? You take oxalate and you add it to acetyl group and you put these together. You just put them together like that? What puts these things together in your cells? What catalyzes these reactions? Enzymes. Enzymes. 99% of the proteins you make are catalytic proteins. They're catalyzing reactions. They're the reason why we run. Literally. Right? So if you had, say, a sugar that you wanted to break down in glycolysis per se, if you think just one enzyme breaks down glucose to pyruvate, no, it's a stepwise procedure, right? There's several enzymes involved in that pathway, right? That process. So would you need those enzymes one at a time? No, if you want to do glycolysis, you need all the enzymes to do glycolysis all at the same time, right? So it'd probably be a good idea to make them all at the same time. Yeah? Make sense to you guys? So they'll make ones involved in a metabolic pathway all at the same time. And they can do this very quickly because they couple transcription and translation. Right? And they do these big, long messenger RNAs that have more than one gene on them. Make more than one protein at a time. So proteins are important, and their structure is important. And how is their structure determined? The messenger RNA tells us what amino acids to put there, right? Each one of those amino acids plays an important role, right? Especially when you have interstrand binding, right? When you make a protein, it's not just this big long string of amino acids, right? It folds back on itself. It'll form alpha helixes, right? Beta sheets, and you get this big globular protein, right? Which is really important, especially if you're an enzyme and you need to grab a substrate and facilitate breaking it into 2 pieces, right? So is your structure important? Oh my God, more than anything. And if you could crack this code, you'd be a gazillionaire. Because scientists are still trying to figure out how cells fold up proteins based on their amino acid sequence. It's still so much of a mystery to us. But the core of it all, too, is you can't get some of these bonds to happen if you don't have this amino acid here and this amino acid here. So what happens if we change one of those amino acids? Is this protein going to look the same? No, we could literally change one amino acid and this protein not work anymore. You say not be able to do glycolysis anymore. That would be really bad news, right? If you eliminated a pathway, something like that. So change, do we really want change to happen? Depends on the change, right? If it means that we can't do something anymore, that puts us at a disadvantage. But what if that change, could that change be good? Could it make this enzyme faster, stronger, resistant to antibiotics? Sure, it happens. So that's what we're going to look at, is now looking at what happens if this code changes. What effect does it have on the proteins that we're producing? Because remember, all of that information goes right back to the DNA. So if there's a change in the DNA, is there going to be a change in the RNA? Yep. Is there going to be a change in the protein? That's the tricky one. Depends. Because we're talking about multiple steps here, right? So what do we mean by base substitution? So when we say base, we're talking about that code, the A's, the G's, the T's, and the C's, the nitrogenous bases. What if we substitute just one of those, right? We put the wrong one in. Some of the mutations that can happen are what we refer to as a silent mutation, a missense mutation, and a nonsense mutation. Other types of mutations that we're going to look at are separate. Frameshift is a different phenomenon that we'll look at, and transposons are different as well. I need to add a slide for us to be able to talk about knockout mutations. They don't talk about it in this chapter in this book. We can induce mutations. We can cause them to happen. The most common way is chemical mutagens. They picked a different one in your book. I want to add, for the next PowerPoint I put up for you guys, a substance known as athenium bromine, which is a specific chemical mutagen that scientists used to use in the lab a lot more. We're using it less and less because guess what? It's a mutagen. It mutates and changes DNA. So do we want to be exposed to that? No. Right? Radiation, and we already touched a little bit on this, right, last time, in X-rays. All of these can change the DNA and therefore change potentially the proteins and what you can and can't do. Mistakes in the copying of the DNA we call mutations. If it doesn't get fixed and it gets passed on to the next generation, they don't die. That's a mutation. They're not always bad, as I said, right? And they're actually the reason why life has evolved and changed on Earth, because these mistakes happen, and sometimes it's good. How many of you guys watched the video of the bacteria mutating across that huge, giant petri dish? Pretty crazy stuff, right? I haven't had a chance to look at it, but I'm really, you know what I'm curious about? What mutation happened, right? Have they sequenced the DNA? Have they figured out, right, what exactly changed, right? That they were able to be resistant to those antibiotics now. And then, of course, what antibiotic did they use? So they can alter the coding portion of a gene, resulting in abbreviating the function of the protein, right? Making some change in it, because remember, it goes from DNA to RNA to protein. So with base substitutions, where you just switch out one base for the other, right? This is not a big change, right? And because of that, sometimes you will get a change in the DNA and you won't get a change in the protein. More often, this is in that third position of the codon, right? Where we've got some wiggle room. So what happens? The DNA changes, so it's still a mutation. We still got to call it a mutation. The RNA has changed, but when the ribosome puts the amino acid there, it's the same one that it always had been putting there. So we call this silent because you don't really see the effect of the mutation, right, on the final product. The protein does not change, but the DNA did change. So we still have to call it a mutation. The next thing that can happen is that we do get a change in the amino acid. And codons that code for amino acids are referred to as sense codons. That's where this name comes from. Missense means you made a mistake, right? Instead of putting the normal amino acid there that's supposed to be there, there's a mistake that's made. A different amino acid gets put there. Could that one amino acid change that protein significantly? We're talking about one amino acid. What's the big deal, right? Could it make that protein non-functional? Absolutely. Could it make it work better? Sure could. Could it make it resistant to antibiotics? Sure could. Just like when you buy a lottery ticket, right? You could win, you could lose. Could go either way. More often than not, you're going to lose. That's why I don't bother buying them. But this happens no matter what. Right? Mutation happens over time. Change happens over time. But of course, we're going to select for the good changes, right? If it means now you can't do something, and that's something you need to live, that cell's going to die, right? It's not going to become part of the evolutionary process. It becomes extinct. But if it's good, right, it's now resistant to an antibiotic, or it's faster than all its buddies, it's going to predominate in the population. The next possibility is a nonsense mutation. Nonsense are our non-coding codons. These are our 3 stop codons. So instead of it coding for an amino acid like it's supposed to, it codes for a stop codon. So what happens when you get up to an intersection and you stop like 4 feet in front of the line? Well, it's a red light. Most people in New Orleans will run into you, yeah. You look in the rearview mirror, right? Decide on whether you're even going to stop at the yellow light, right? Because let me put it that way, right? If you stop at a yellow light, that's a premature stop as far as everyone in New Orleans is concerned, right? You should actually go through that, right? Not really. Okay, so if you're a protein, right? And the ribosome stops before it's supposed to. It doesn't even make all of you, right? He stops, 10 amino acids short. Okay. Is that really going to be okay? You're missing 10 amino acids at the end of your protein. I mean, if you didn't need them, you would never make them to begin with, right? But probably you kind of need them, right? So what's the chance that stop codon basically stopping prematurely before you're supposed to is going to be a good mutation? Not good, huh? Right? If you had to lay down your bets, right? you would say, I'll take the silent one, right? So let's look at an actual example of this. So this is the wild type. This is what the sequence is supposed to be. This is what most of the population is. That's what wild type means, right? The most prominent within the population. So for a particular protein, it has methionine, and it would not be this short, y'all. It'd be like thousands of amino acids long. This is just for an example, right? So it has the start, methionine, it's tryptophan. I have no idea what ILE is for. I don't know all the 3-letter abbreviations of the amino acids. And this is arginine, and then here's a stop codon. So that's what it's supposed to be. Here, in this example, notice right here in the 3rd position, it says TA. But if you look up here for arginine, it should be CG, right? So a mutation happened when they were copying the DNA. So right at the DNA level. So of course then it causes a change in the RNA. But, remarkably, no change in the amino acid. We're still putting arginine there. So that's what we call a silent mutation, right? You don't see a change in the product, in the protein, but there is in fact a change in the DNA. So it's still a mutation. This one, it changes it in that first position, right? The DNA gets changed. So now the codon changes significantly that we get a different amino acid than we're supposed to. So there's serine instead of arginine. What effect is that going to have on the protein? Is that going to be good? Maybe. Is it going to be bad? Maybe. Is it going to have no effect at all? Possibly. Right? It could be any of those things. And variance within it, right? Could be really good. Could be have no effect at all. Could be bad in that you can't do something that you were able to do before. Now you have to rely on maybe other things in the environment to provide it for you. Or it might be lethal. And in that case, it's not going to continue on, right? This next one, notice methionine then stops at the yellow light. It probably gets rear-ended because what's the chance that just methionine is going to be a functioning protein? Yeah, no. 0. Very unlikely, right, when we stop before we're supposed to, that this is going to result in a mutation that's ever going to be good. And again, how bad it is really depends on what protein are we talking about, right? How crucial is that to the functioning of that cell or that organism? The next one is where it gets interesting. So the next is frame shift. So as the name says, right, we're shifting the frame. This is the problem with how we read messenger RNA to translate it into proteins. We read it in groups of 3, right? So if we, or it, right, removes a nucleotide, or what are they doing in this example? They're adding, aren't they? Yeah, they're adding. So in this case, they're adding a CG. So it's fine up until that point, right? Methionine, lysine, right? But right here, it's supposed to be glycine. But because they added this amino acid, it is now arginine. But the problem is the ribosome doesn't know you added a nucleotide, right? It's going to keep doing like it always does. 3, 3, 3, 3. It keeps reading in groups of 3. So by having inserted this, we just shifted the code over, right? So we messed up everything after that. So the best example I can give to students that they usually remember, because you guys are still in some classes doing Scantron tests, right? So imagine you're taking a Scantron test, right? And you don't know, right? You're just like kind of stuck on number 10, right? And you're like, oh, I'm just going to skip it and come back to it. But then on your Scantron, for number 11, you answer in number 10 spot. It's not good, right? Then you keep going down your test, and then you realize, oh, they don't match up, right? So if you hand that in, what's the likelihood from like 10 on you're going to get those right? 0. Yeah, not good, right? Because they don't match up. Everything from that point, from when you made that mistake, gets messed up, right? Everything is shifted, you see? Yeah, probably failed your entire test. Same thing if you answer, like, you know, somebody, and I'll tell you in honesty, like, teachers don't do this on purpose, but sometimes you have, like, 4 C's in a row, right? And then you do 5 C's. And again, now you've put too many, and now the whole rest is shifted again. So imagine this in your DNA, though, right? Again, it's going to probably mess up everything. And even worse, because of that, it tends to put a stop codon before it's supposed to. So not only do you have a whole string of amino acids that are wrong, you're stopping even before you're supposed to. Frame shifts are the most damaging. You could pretty much end up with a non-functional protein. Yeah, but because that one isn't an isolated problem, right? Unlike before we talked about just switching out one base. In this case, if you add or remove a nucleotide, you change everything from that point forward, just like on your Scantron test. So this is an old example I used to type out for people that might help as well. And I didn't put the slide in because I think you guys would understand if I build it as I went. So this is an example of frame shift, but maybe in terms that we would understand. So remember, it reads in groups of 3. So I'm going to write this in groups of 3 letters that actually make up a word, right? So the dog ate the cat. Makes sense, right? No problem there. But I'm a ribosome now, right? And I can only read in groups of 3 letters. But somebody decided they were going to add an A to the beginning of this sentence. But I'm a ribosome. All I know how to do is read in groups of 3, right? Get my little transfer RNA, so let's do groups of 3. So now I go TH, right? And then I go to the next one, right? EDO, GAT, ETH, EC. Wow. Does that make any sense? No. We just messed up that whole sentence, didn't we? It shifted the whole sentence by starting out with adding an extra letter. Or in the case of DNA, adding an extra nitrogenous base. Right? An extra base. We messed up all the codons. What happens if we add, take away? Let's take away the T. Remember, we're a ribosome, right? So we're going to go HED, OGA, TET. Messed up the whole sentence again, huh? There's one exception to frame shifting, where you won't get a shift in the frame. If you add a whole codon, right? So I added big. And remember, these are representing codons, right? Each one of these words is a codon. So in this case, if we were talking in terms of DNA, right, I added 3 nucleotides, right? In the RNA, there's a whole another codon, which codes for what? An amino acid. So I just inserted an amino acid in this sentence. Inserted a word, inserted an amino acid. I didn't create a shift, did I? Because I did it in between codons. These are extremely rare, but it can happen. This is the only time we're adding or removing nucleotides, because I could also just remove, right? Right, the dog ate cat. English teacher wouldn't be too happy about that, but right? I could do it. What effect would that have on the protein, though? Would that be as damaging as a frame shift? You think so? If I added or removed an amino acid?
That's one of the possibilities. It could.
It could change it. It could be good. It could be bad. Right? In this case, there could be a good. Where with a frame shift, we usually screw up everything from that point, and most of the time we're going to stop before we're supposed to. More often than not, it's going to be a bad thing. This is the one exception, like I said, to adding or removing is where you add or remove a whole codon. So therefore you're adding or removing an entire amino acid, and you're preserving the rest of the sequence. You're not creating a shift. Yeah, and if I remove, you're shorting it, yeah, I mean, there's the potential that that could be bad. But you could come out with something good. Make sense to you guys? Putting it in words instead of, nucleotides. And does that help a little bit? Okay. So other things that can happen are whole sections can move. And this kind of links into transposable elements that we'll talk about later, where whole segments of the chromosome, right? So say this section pops out, and in this case it flipped over and inverted. Or hopping from one chromosome to another. These usually aren't good. Right? Big major changes like this usually don't result in anything good. So you guys remember what enzyme we've already talked about has the ability to proofread as it's making the DNA? What copies DNA? Polymerase. DNA polymerase, right? Because there's an RNA polymerase that makes RNA, but DNA copies DNA. And so changes and mutations, right, at that level of copying the DNA are relatively low because it can proofread its own work. But as you guys know, right, we have the ability to do the same thing, right? You have the ability to proofread your work as you're doing it. Do you always catch every single little mistake? No, right? And neither does DNA polymerase, although I think he probably does a whole lot better than we do. Right? But it happens, and that's why change happens, because they do make a mistake and don't detect it, don't fix it. But we're looking at a rate and probably of 1 in every billion that they do, that they make a mistake. So other things that can cause mistakes to be made in the copying is chemicals that induce changes in the nitrogenous bases. We refer to this as induced mutations, right? Because they're caused to happen, and in this case by chemicals. And of course this happens more often than just spontaneous mutations where DNA polymerase just makes a mistake. So scientists actually will sometimes purposely do this to see what happens. A lot of times we test chemicals to see if this happens, and obviously if it's a chemical we want to take internally or use on our bodies, or if it changes DNA, we certainly don't want to use that, right? If it mutates or causes changes in DNA, we don't want that. So we know that there aren't huge differences between these nitrogenous bases. Some of the chemical groups that bind with the complementary base, if you change those, it'll bind with a different base. So we know adenine is supposed to bind with what? Thymine, right? If a chemical mutagen changes its functional group here, you can make this weird nitrogenous base known as hypoxanthine, and it will bind with cysteine instead of thymine. You can modify cysteine where it'll turn into uracil, which will then bind with adenine. And of course, uracil's not even supposed to be in DNA, right? What does that do in there? I expect you guys to memorize these? Oh God, no. Right? But understand that we can modify the bases such that they won't bind with the correct base anymore. So then you get this switch. Okay, so we'll pick this up next time. And I'm going to add some more slides, probably in a separate PowerPoint, and post them for you guys later today.